Dear Grade 12, I am Mrs. V. Ramlal. Welcome to the Junior Tucky Winter School for Grade 12s. There will be a series of videos which will be live streamed in the next few weeks on Junior Tucky Online. Please use this material as revision and also look at all the Grade 12 content in Life Sciences on Junior Tucky Online. In this video, I will be looking at the question 3 of the Supp 2022 Paper 1. Question 3. Question 3.1 and 3.2 are two different sections and two different parts. So 3.1, it asks you to describe the role of auxin in phototropism when a plant is exposed to unilateral light. That is for 5 marks. 3.2, the Fsh test is sometimes used to determine the cause of infertility in females. Now we know FSH stands for follicle stimulating hormone. The levels of FSH usually indicate the number of follicles in the ovaries. If the number of follicles are low or depleted, the pituitary gland will secrete more FSH. So, an investigation was conducted to compare the average FSH levels in 4 different age groups. So, the procedure was done as follows. 1,000 females were asked to participate. There were 250 each of the 4 age groups. The females were all healthy and not using any hormone-based contraceptives. Their blood FSH levels were measured on day 3 of the menstrual cycle for 5 cycles. The average FSH levels in their blood were calculated per age group, and the results are shown in the table below. So in the table you have the age groups 20 to 32, 33 to 35, 36 to 40, and 41 to 50. And here is your average FSH levels. Right, what are they asking you? 3.2.1. State 2 factors regarding the females that should have been kept constant during the investigation. 2 marks. 3.2.2. State 2 ways in which the reliability of the results was ensured. 2 marks. 3.2.3. State 1 conclusion that can be drawn from the results. 2 marks. 3.2.4. Explain why the estrogen levels may remain low in the blood of the females in 41 to 50 age group. 3 marks. And 3.2.5. Explain why females that were using progesterone-based pills were excluded from the investigation. 3 marks. And all of this adds up to 12 marks. Right, let's do some revision to help us answer question 3. Right, phototropism. Diagram A and B shows how different shoots are exposed to and how they respond to different types of light. So let's look at B. B, the shoot is exposed to light from one direction, so we know that is unilateral light. This is shoot A is exposed to light from all directions, uniform light. Now, we know auxin is produced in the tip. Because auxin now is exposed to uniform light, the auxin will move down evenly on either side of the shoot, and because there's equal amount of auxin on either side of the shoot will grow straight upwards. Diagram B is exposed to unilateral light. The auxin is produced at the tip, but we know auxin is now sensitive to light, so it will move to the shady part. And because there's more auxin here on the shady part or on the darker side of the shoot, there will be more cell division, more cell elongation, growing and bending towards light. So it's these cells here that will grow and bend towards light, and that is phototropism. Right, let's look at the menstrual cycle. Let's look at the part where they talk about FSH, and let's quickly revise. Now, the menstrual cycle. The menstrual cycle takes place over 28 days, right? And in this diagram, 0 to 7, that is where you have your menstruation that's taking place, which is the sloughing off of the endometrium lining. Now, as soon as the menstruation has taken place, the pituitary gland will produce FSH, follicle stimulating hormone. And this hormone causes the follicle to develop from a follicle to a primary to a secondary to a Graafian follicle. And as this follicle is forming, the follicle is producing estrogen. And the estrogen is increasing because the follicle size is increasing. What does estrogen do? Estrogen makes the endometrium lining more thick, vascular, and glandular for implantation. Now, on the 14th day, we know ovulation will take place, which is when the Graafian follicle will burst, releasing the ovum. And the remains of the Graafian follicle will now form the corpus luteum, which will now produce progesterone. And now progesterone will carry on making the endometrium lining more thicker, more vascular, more glandular for implantation. So here we can see the link of FSH and estrogen. So we're only going to be talking about in the question about FSH and estrogen. We don't need to talk about progesterone at all because there's no corpus luteum and there's no luteinizing hormone that is being mentioned. Right, after we've done some revision on work that you need for question 3, let's answer question 3. Right, 3.1. 3.1, describe the role of auxins in phototropism when a plant is exposed to unilateral light. Now, I've already discussed this in our revision video or revision slide. So 3.1, we know auxin is sensitive to light. So auxin will move away from the light, and so we know auxin will move to the shady part or to the darker side. And because there will be auxin moves to the shady part, there'll be more auxin on the shady or the dark side. And the more auxin on the shady or the dark side, that will cause more growth to occur, and more growth will be stimulated, which will cause the growth to be occurring faster on the darker side, and this will cause the stem to grow and bend towards light. That's what was required for 5 marks. Right. 3.2. The FSH test, or the follicle stimulating hormone test, is sometimes used to determine the cause of infertility in females. The levels of FSH usually indicate the number of follicles in the ovaries. If the number of follicles are old, the pituitary gland will secrete more FSH. So, an investigation was conducted to compare the average FSH levels in 4 different age groups. The procedure was done as follows. 1,000 females were asked to participate, 250 in each of the 4 age groups. The females were all healthy and not using any hormone-based contraceptives. Their blood FSH levels were measured on day 3 of the menstrual cycle for 5 cycles. And the average FSH levels in their bloods were calculated per average age group. Right, now we've already discussed the table. Let's answer the questions. 3.2.1. State 2 factors regarding the females that should have been kept constant during the investigation. This took me some time to think about. And I was thinking, what is it that the females must keep fixed or constant that could affect their average FSH levels? And that's easy. They must have their same dietary intake. So the food that they take can also influence their hormone production, the level of the hormone production. So same dietary intake. And then what else regarding the females must be kept constant so it doesn't affect the investigation? They must have obviously regular menstrual cycles. Obviously, if they don't have a regular menstrual cycle, then they cannot produce FSH and the other important hormones. Right. Now, 3.2.2. State 2 ways in which the reliability of the results was ensured. Now, reliability was ensured. Ensured means it's already done in this text, in this investigation. You have to select it. Now, with reliability, it's usually increased the sample size. Here you can see 1,000 females were used. 250 in each of the 4 age groups. That is one of the ways in which reliability was ensured. Then the next one, we can repeat the investigation, but here the investigation was not repeated. Here the menstrual cycle, the measurements were taken for 5 cycles. So measurement was done for 5 cycles. So that is how reliability was ensured. 3.2.3. State 1 conclusion that can be drawn from the results. Now if you look at the age group, as the age group is increasing, okay, you can see the average in younger women, the average FSH levels are less because they have a lot of follicles. Older women, they have fewer follicles, so then their pituitary gland will produce more FSH. And now, can you see they have younger women and older women, and they have lower amounts and higher amounts. So you have to actually compare them. You have to compare older women to younger women. So I would have actually said that older women have a higher FSH levels than younger women. You could have also said older women have a lower average FSH levels than younger women. Sorry. Let's do this again. So you could have said that younger women have a lower average FSH levels than older women. You could have said older women have a higher average FSH levels than younger women. So you could have said it either way, older versus young or young versus old. Anyway, but you have to compare the two. Right. Now that we've got our bearings on that question, sure. So remember, always compare the two and always have average FSH levels in your answer. 3.2.4. Explain why the estrogen levels are low in the blood of the females in 41 to 50 age group. Now, we know the graphene follicle secretes estrogen, right? And as you get older, the woman will be reaching menopause, so they will produce fewer follicles, and so their estrogen levels will be lower. Okay. 3.2.5. Explain why females that were using progesterone-based pills were excluded in the investigation. Now, this is a very important and a nice question. I like how they brought this question in. So we know a high amount of progesterone. They're going to be on the contraceptive, so there will be high amount of progesterone in their body. That will inhibit FSH, and the pituitary gland will no longer secrete estrogen follicles, and there'll be no estrogen produced. And we want to measure FSH and estrogen, and that will affect our results, and that will affect the validity of the experiment. Right, that was question 3. That was quite a tough question. Right, practice this question. Make sure you master this question. Make sure you can open your mind to answering a similar type of question in your prelim or your final exam. Right, let's look at 3.3. 3.3, you are asked to describe the development of the placenta and the umbilical cord from the time of implantation. 6 marks. So this is still reproduction. 3.4, the diagram below represents part of the human ear with the middle ear infection. Right, so there's your diagram. There's A, B, C, D, E, and F, and then the middle ear, there's an infection. 3.4.1, they want you to identify part B. They want you to identify parts D. Can you see it's all 3 of these parts? 3.4.2, state one function of part A. 3.4.3, how middle ear infection can affect hearing. 3.4.4, describe the role of the Eustachian tube, which is this tube here. 3.4.5, name the small device that is used in the treatment of middle ear infection. 3.4.6, write down the letter of the part where the small device named in 3.4.5 is inserted. And 3.4.7, describe how part E is involved in maintaining balance where there's a change in speed and direction of movement of the head. Right, let's do some revision before we answer this question. Right, we are going to do the adaptations of the ear. We'll do the outer ear, middle ear, and inner ear. So adaptations is how the structure helps the function. Some of this content will help us with answering the questions 3.3 and 3.4. Right, this part here is the Pano. The Pano is flexible because it's made up of cartilage. It's funnel shaped, it has these grooves, and it's used to trap and direct the sound waves, and it collects the sound waves and directs it to the auditory canal. The auditory canal is this long part. It's long and hollow to allow the sound waves to travel through, and then it will hit the tympanum or the tympanic membrane. Some other information. The auditory canal has cilia and ceruminous glands. Ceruminous glands produce cerumen, and cerumen is wax, but we cannot call it wax. And wax has a pungent smell to prevent insects from entering the ear. Right, so that's the outer ear. Now let's go to the middle ear. There's your tympanum or your tympanic membrane. You can decide whichever word you use, but do not use the word eardrum. When the sound waves hits the tympanum vibrates. Now attached to the tympanum is the 3 ossicles, the hammer, the anvil, and the stirrup. The hammer, anvil, and stirrup, you can remember it as has, H-A-S, has. So these are 3 tiny bones, tiny ossicles, and they hollow. So when these 3 bones vibrate, these bones will also amplify the sound and amplify the vibrations as well. So the hammer, when it vibrates, it causes the anvil to vibrate. When the anvil vibrates, it causes the stirrup to vibrate. Now, that's what the ossicles do. When they vibrate, they also amplify the vibrations. Now, this is the Eustachian tube. The Eustachian tube is connected to the middle ear and from the pharynx. And the Eustachian tube equalizes pressure on either side of the tympanum. Right, that was the outer ear and the middle ear. Right, let's get to the inner ear. The inner ear, there's your oval window and your round window. From its name, one window is oval and the other is actually round. The oval window will then transmit the vibrations to the inner ear. which is the cochlea. The round window absorbs excess pressure from the sound waves. Right. There's your semicircular canals. Your semicircular canals are responsible for balance. There's your cochlea, which converts the stimulus into an impulse and transmits the impulse to the cerebrum to interpret hearing. The auditory nerve transmits the impulse to the cerebrum for hearing to be interpreted. So, this is now the inner ear. This will help us with our question on the ear. Right, this information here is going to help us on question 3.3. We're just going to quickly revise the placenta and the umbilical cord. So we know that the trophoblast from the blastocyst will form the chorionic villi, which are these structures here. The chorionic villi grows into the endometrium, and that's when implantation takes place. Now, the embryonic blood vessels and the endometrium make up the placenta. So it's basically made up of the mother's blood vessels and the fetal blood vessels and a sinus. The two blood vessels will never touch. There's actually a sinus, and substances will enter by diffusion. Now, connected to the placenta and the fetus is the umbilical cord. In the umbilical cord are two umbilical arteries and one umbilical vein. The vein carries oxygenated blood and the arteries carry deoxygenated blood. Right. This will also help us now with the answering of question 3.3. Right. Let's get down to answering the questions. 3.3. Describe the development of the placenta and the umbilical cord from the time of implantation. So, we know after implantation, the chorion will develop the finger-like outgrowths. What are these called? The chorionic villi. The endometrium, together with the chorionic villi, forms the placenta. The placenta is made up of the embryonic and the maternal tissues or blood vessels. The umbilical cord develops between the fetus and the placenta, and it consists of a hollow tube. What's in that hollow tube? The umbilical artery and the umbilical vein. Up to 8 to 12 weeks, that development of the embryo will now form tissues and organs and is now called a fetus. So, as you can see here, it's only 6 marks, but any of those 6 marks. So, you must know the formation of the chorion, the chorionic villi, what makes up the placenta, the cord, what is found in the cord, the umbilical vein, the umbilical arteries. It's very, very important. I would recommend that you learn this question. I'm seeing this question coming out too frequently, so it might come out in your prelim or your final exam. Right. Let's get down to 3.4, the question on the ear. Right. The diagram below represents part of the human ear with the middle ear infection. 3.4.1. Identify part B. Now we already did this. Part B is the auditory canal. Identify part D. Part D, we know. Can you see it's not asking for the hammer, the anvil, or the stirrup? They're asking for all of them. Those are the ossicles. 3.4.2. State one function of part A. Part A, we know, is the pinna. So what is the pinna going to do? Trap and collect and direct sound waves to the auditory canal. 3.4.3. Explain how middle ear infection could affect hearing. Now, if there's mucus here, then that will cause the vibrations, few vibrations from the tympanum to go to the ossicles. And if there's fewer vibrations will then go to the cochlea. And then fewer vibrations will be interpreted, and that will cause a problem with hearing. So, that's what I would say, fewer vibrations. The memo also might say no vibrations. So you have an decision to make, whether you're going to say fewer or no vibrations. If you're going to say fewer, make sure you carry on through and use the word fewer and less throughout the answer. If you're going to say no vibrations, make sure you'll say no pressure waves. The organ of Corti will not be stimulated, and this will not stimulate the cerebrum, and no hearing loss will occur. So make sure whichever one you're going to use, fewer vibrations or no vibrations, you carry through the same answer throughout the answer that you're writing. Okay, I hope that you understand carefully. I hope you understand properly. Sorry. Then, describe the role. 3.4.4. Describe the role of the Eustachian tube. We just learned the Eustachian tube equalizes pressure on either side of the tympanic membrane or tympanum. 3.4.5. Name the small device that's used in the treatment of middle ear infections. Yes, we know for babies that will be grommets. Write on the letter of the part where the grommet is inserted The grommet is usually inserted into the tympanum, and the tympanum or the tympanic membrane is letter C. 347. 347. Describe how part E, which is the semicircular canals, is involved in maintaining balance when there's a change in the speed and direction of movement of the head. 4 marks. This is quite easy. They don't give you a scenario. They just talk about the cristae. So what you're required to say is the cristae in the ampulla in the semicircular canals. So the cristae are in this ampulla, in these canals, these semicircular canals. They will pick up or detect changes in speed and direction, so they will be stimulated. This will generate an impulse, which will be transmitted via the vestibular branch of the auditory nerve, which will go to the cerebellum, where the impulse is transmitted, and then impulses will be sent via the motor neuron to the skeletal muscles to restore balance. Make sure in your answer here you always have stimulus, impulse, interpreted. That's always the order for the eye or the ear or balance. Always have stimulus, impulse, interpreted. Okay, so this was a nice question, not too difficult. It was a question that we could do answer after we did our revision. Right. Let's go to 3.5. Let's read 3.5. Type 1 diabetes mellitus is caused by pancreatic cells which cannot produce insulin. Type 2 diabetes mellitus, the pancreas does secrete insulin, but the body is resistant to the effect of insulin. To diagnose if a person has type 1 or type 2 diabetes mellitus, the following tests may be conducted. The blood glucose level is tested after fasting. The person is not allowed to eat or drink for 6 hours before the test. If the blood glucose level is above 126 milligrams per deciliter, the person is diabetic. The person is then given a glucose solution to drink, and the blood glucose level is tested after 2 hours. The blood is also tested for the presence of GAD antibodies. The presence of GAD antibodies will indicate that the body is destroying the insulin-producing pancreatic cells. Now, if the pancreatic cells have been destroyed, then there's no insulin that's going to be produced here. Right. The levels of the C-peptide are measured. This is a substance produced by the same pancreatic cells that produce insulin. If the levels are below 0.3 ng per milliliter in the blood, the person is a diabetic. Right. Now, let's see what they want us to answer. 351. Name the cells in the pancreas that secrete insulin. 1 mark. 352. Explain which type, 1 or 2, of diabetes mellitus will be indicated by the A. Presence of the GAD antibodies in the blood. 3 marks. So you must say which type of which type of diabetes, 1 or 2, and explain. B. Explain which type 1 or 2 diabetes will have the C-peptide levels below 0.3 ng per milliliters. That's also 3 marks. So which type of diabetes, and explain. 353. Explain the insulin levels in the blood of a type 2 diabetic 2 hours after drinking the glucose solution. 3 marks. And the last question, 354. Give one reason why a person with untreated diabetes mellitus is expected to be tired all the time. Now, as you're reading this, and as I'm reading this, this is a difficult question. It's been a long time since I've seen such high-level questions. So there's your level 4 question. Right, let's revise some work before we answer this question. Type 1 diabetes is an autoimmune disease. So your body cells, your white blood cells are fighting off the pancreas. Type 2 is insulin resistance. Type 1, your pancreas cannot produce any insulin. Type 2, it does produce insulin, but your body cannot use it. Type 1, diet, exercise, or other lifestyle changes cannot reverse the condition. Type 2, diet, exercise, and oral medications can help the body produce insulin more effectively. Here, the beta cells have been destroyed. Type 2, the beta cells will deteriorate over time, and this person for type 1 is dependent on insulin for the rest of their lives. Here, type 2, the insulin injections may be required at some point. Right, we require this information to answer the question that we just read. Right, our last question for the sub paper. Now, 351. Name the cells in the pancreas that secrete insulin. So we know it's the beta cells, and we know the beta cells are in the islets of Langerhans. If you said the beta cells of the islets of Langerhans, I'm sure they would still accept that. 352. Which type of diabetes would be indicated by the presence of GAD antibodies in the blood? GAD antibodies indicate the blood is destroying the insulin-producing pancreatic cells. So, if there's no insulin, right, no insulin produced and the cells are destroyed, that can only be type 1 diabetes, because you say that no insulin is produced because the presence of these antibodies indicate the pancreas cells are being destroyed. Right, explain which type of diabetes is indicated by the C-peptide levels at below 0.3 ng per milliliters. Now, again, if the levels are below 0.3, that means it's a substance produced by the pancreatic cells. If the levels are below, the person is diabetic. That will also be type 1 diabetes. And if the person has a lower level, that indicates also that there's no insulin because their pancreas is being destroyed. This is a tricky question. Don't doubt yourself. Don't always have to write 1 or 2, type 1 or type 2. And if you had type 1 and type 1, you probably would have doubted yourself and said, nah, it can't be the answer. Don't doubt yourself. The answer is here in the explanation. So stick to what they give you. Right. 353. Explain the insulin levels in the blood of a type 2 diabetic 2 hours after drinking the glucose solution. Now we know after you drink the glucose solution, what's going to happen? Your insulin levels will be high. Why? Your blood glucose level is high, and that will cause your pancreas to carry on secreting insulin to try and remove the excess glucose in your blood. Alright, simple, straight to the point. 354. Give one reason why a person with untreated diabetes mellitus is expected to be tired all the time. Now, the glucose is supposed to be used by the cells, right, for cellular respiration. But now the problem is the glucose is staying in the blood and it's not used by cellular respiration. And as a result, there's less glucose for cellular respiration, and that is why the person is tired all the time. I really like doing this question. I really enjoyed this question. Well done to the examiner for putting a good level 4 question in. But please try to open your mind and answer this question. See that everything is in the text. Don't be scared. Use your knowledge that you have in the text and answer from there. Don't doubt yourself. Thank you for joining me and watching the question 3 of the Supp paper. Join me and watch all my other videos as well.